Matrix, welcome to your show. It's Funny Your Time, 6 o'clock, Wednesday. You know what it is, Life Sciences. You're hanging out with Looney and Aslam. How are you, Aslam?
Looney, you're becoming tuning, huh?
Yeah, you know, I gotta just...
Hang in there.
What are we doing? I'm good, yes. What are you doing?
Continuing from last week, we stopped with DNA. We're moving to RNA, the other nucleic acid, and protein synthesis.
All right, Matrix, you heard it, RNA and protein synthesis. I hope you guys do enjoy the show. Remember, if you want the notes, the videos, and the schedules, you can get them on learn.mindset.co.za. Follow us on Facebook and on Twitter at LearnExtra. And remember to show your love, seeing as though it's the month of love. We at Mindset are trying to, you know, show the love and show you guys some love. So all you need to do is post your pictures on our Facebook page with either someone special or something special. So you have to be in the picture with that thing. So if you like your couch, you must just take a selfie with your couch. Tell us why it means so much to you, and you might just see yourself on TV, guys. So make sure you do that. Make sure you hashtag your picture, show your love, comment below why it means so much to you. As simple as that. But remember, you don't have to show love in February only, because we have other months. So you must just continue all year, every year, all day, every day. Aslam.
Are we ready, Looney?
Yes.
Good, good, good. Selfie is a big word that just hit the market with the storm.
Yes, it did.
Yeah. Welcome back, Mindsetters, grade 12s. I'm hoping by now you've settled into the work and you're getting used to the volume at school, the homework and assignments and everything else that goes through. We're moving. February is already halfway down almost. End of this month, you're going to start writing tests, and then it's the exams, and the term is over. So get ready, and let's start moving with what we are talking about. RNA and protein synthesis, as we said earlier, and what are we looking at specifically in this lesson? We are looking for, or at rather, the location of RNA. Where do we find RNA? Secondly, we want to look at the structure of RNA, where in terms of that it's also a single strand. Not also, it is a single strand. It also has phosphate. It has sugar, but the ribose sugar. And it has nitrogenous bases, but more about that a little bit later. We want to compare DNA and RNA. We've learned the structure of DNA last week. Now we look at RNA. So we must be able to say this is what DNA has, this is what RNA has. We must define what protein synthesis is, and that's simple English. Protein and synthesis, what do they mean to us? And lastly, we want to describe the involvement of DNA and RNA in protein synthesis. Obviously, when we describe the involvement of these two molecules in protein synthesis, what we are doing in essence is describing protein synthesis. Also, in between, we must also talk about the functions of RNA as well. So that's what we're going to be doing today. Let's see. Let's take it from there. Obviously, before we start with the pattern now, remember we say we give you the challenge question. The challenge question is already posted, Luni?
Facebook.
Yeah, it's on Facebook already. And the question is, the first 7 triplets of nitrogenous bases that form part of a gene coding for one chain of the hemoglobin protein that makes up red blood corpuscles in humans is shown below. So they're giving you the DNA template. These are the base triplets, and they give you the number. This is number 1, 2, 3, 4, 5, 6, 7. They want to know how many of the following are coded for in this DNA template. How many of the following? Firstly, how many nitrogenous bases are there? Secondly, how many different types of transfer RNA molecules? And lastly, how many amino acids will this particular molecule code for? And then B, write down the messenger RNA sequence from triplet number 4 to triplet number 6 for the DNA template above. In other words, they've given you the DNA template above. You must give them the messenger RNA that will form there. And C, using the table below, determine the amino acid sequence coded by triplet number 4 to triplet number 6. What's important here is to look at this table and see what they are talking about here. Look at what they're asking you for here. They're giving you a code, but they're giving you the code for the anticodons on transfer RNA. Very important to look at that. Sometimes in this table like this, they will give you the code on DNA. Sometimes they will give you the code on messenger RNA, and this one is giving you the code or the anticode on transfer RNA. And with this, they're giving you the amino acid that this particular code will code for. In other words, these are transfer RNA, and these transfer RNA molecules, for example, CUC, will carry glutamate. Now, remember, mindsetters, you do not need to memorize a table like this. If a question is given like this in an exam, they would always have to give you the table, and all you are doing what we call a simple matching exercise, but you need to apply your mind to do this. It's not a straightforward A-B matching. You need to apply. You need to know your story on how protein synthesis works, what is base pairing to get this right. Okay? So that is our question for today. Challenge question, that is. Good. Now let's summarize what we are talking about. The first thing is the structure of the RNA. And there we have the structure of the RNA. What better way to learn a structure than with the diagram? There's nothing better to teach you about a diagram, than, about a structure, than looking at the diagram. Many of us want to learn a structure by learning chunks of information. RNA has this, RNA has that. Instead of just looking at a diagram and using the diagram to explain the structure. So first of all, you can see that RNA is made up of a single strand. So it is single-stranded. Secondly, it is made up of phosphate, sugar, not any sugar, but a ribose sugar, and nitrogenous bases. Now let's look at the nitrogenous bases shown from the bottom up. Guanine was in DNA, adenine was in DNA, cytosine was in DNA, and what do we have here? We have another nitrogenous base called uracil. Did we have uracil in DNA? No, it was at cute girl. Adenine, thymine, guanine, cytosine. Now here, which one is missing? We have the A, adenine, we have the C, cytosine, we have the G, guanine, but we do not have the T, thymine. So what can we deduce from that? We can make the inference that uracil has replaced thymine. So in other words, in RNA, there is no thymine. Wherever thymine is supposed to be, that is replaced with uracil. We still have adenine, we still have cytosine, we still have guanine. So again, just to recap, a single strand has phosphate, ribose sugar, it has the nitrogenous bases, adenine, guanine, cytosine, and uracil, which we did not hear about in DNA. Good, guys. So that's the taken care of the structure. Can you see what I'm trying to explain? Looking at a diagram, you simply explain that. So in your mind's eye, you close your eyes and you think of the diagram, and on paper, if they have to ask you to describe the structure, remember, if they ask you to describe, you can't draw the structure. If they say draw, you must draw. When they say describe, you must describe. So in your mind's eye, you picture this diagram and you explain it. So this diagram is taken from Life Sciences for All, right? Okay, the next thing is, what are the different types of RNA? And notice in DNA, we only spoke about mitochondrial DNA and the DNA that we find in the nucleus. Also, I mentioned to you that we also have chloroplast DNA, but for our syllabus in grade 12, we worry about the two, mitochondrial DNA and the nuclear DNA. Here, we have three types. We have messenger RNA, transfer RNA, and ribosomal RNA. The two that are really prescribed for us are these two. Messenger RNA is found, or formed rather, in the nucleus. its function is to copy the code from DNA. This normally happens when DNA is making a copy. Last week we spoke about DNA replication. That's when this happens. Transfer RNA, by the way, it's found in the nucleus and it moves to the cytoplasm. Moves to the cytoplasm. Transfer RNA, on the other hand, is found in the cytoplasm. And its job or its function, it acts as a taxi. T for taxi, T for transfer RNA. Its job is to pick up amino acids, and take, I'm not going to write again the amino acids, take them, I'll write them, to the ribosomes. And ribosomal RNA is thought to have a function in the manufacture of the ribosomes themselves, and other books talk about the ribosomal RNA that helps to attach messenger RNA to the ribosomes. Fortunately for you, you don't have to go into that much detail. You just have to know that there are these three types of RNA, and we're more interested in these two. I'm hoping we're on the same page, guys. Good. Now a summary of the process, protein synthesis. Protein synthesis is made up of two chief stages. One is called transcription. It takes place in the nucleus, and translation, which takes place in the ribosome. Now, English is sometimes a very easy language. Sometimes it can be hard, but sometimes it's easy. Transcription comes from the word to transcribe. Oops, what happened there? Okay, let's go back to that. Transcribe. I am going on to the top there, sorry. Transcribe. Transcribe means to make a copy. Notice I'm stressing this. When we spoke about messenger RNA earlier, I gave you the function of messenger RNA, okay? And we said that the function of messenger RNA is to make a copy of DNA. So obviously, now you put that together with what we say transcription is all about. Transcription is that process where, while DNA replication is taking place, messenger RNA forms there. And enzymes are involved in this process. Okay? Messenger RNA then leaves the nucleus and gets attached to the ribosome. I'm not going into too much detail there. That's all you need to know. And guys, that's all you need to explain in transcription. But obviously, you're going to add some flesh as we're going along. But we're going to add that flesh later. Translation, on the other hand, happens at the ribosome. Now, what is it? Where did we end? The messenger RNA was attached to the ribosome. How was it attached to the ribosome? Such that it was exposing the base triplets. Each of these base triplets that are giving the code are called codons. There by messenger RNA. In the meanwhile, transfer RNA was found in the ribosomes, in the cytoplasm. And they pick up these amino acids, different amino acids, and they move to the ribosome, such that they combine with the messenger RNA according to the code provided. And therefore, the transfer RNA base triplets, the three nitrogenous bases, are now called the anticodon. Codon, anticodon. Okay? And what happens there then, the codon-anticodon combination and the amino acids that are next to each other, adjacent amino acids. So transfer RNA, here's transfer RNA 1, here's transfer RNA 2. They're coming together and they have amino acids. The amino acids combine. When the amino acids combine, water is released. This is known as dehydration synthesis. And we can then call them a dipeptide. And so on, the amino acids will combine, tripeptide, polypeptide, protein, more than 50, right? So we're talking about peptide link, dipeptide, tripeptide, polypeptide, anything more than 50, and that will be called a protein. And I told you water is released. This is dehydration synthesis, and the enzymes control this, and the end result is protein. Now let's go back to what we said originally. What did we have to do? We needed to define, we needed to explain what is meant by this thing called protein synthesis. And in our English language again, protein, we know, synthesis, to synthesize, to make. So protein synthesis refers to the manufacture of proteins. Okay? And the two stories there that we spoke about are transcription and translation. Transcription, to make a copy. Now let's look at this translation part. Now here was a code. We got a code. DNA provided a code during replication. Messenger RNA steals this code, moves out of the nucleus. Why does this happen? DNA has to give the code because DNA has the genes to make a protein. But DNA can't leave the nucleus. The DNA stays in the nucleus. So what does it do? It gives this job to messenger RNA. It releases the story. Messenger RNA takes the code and goes to the ribosome, exposing the code. So in other words, it has stolen the formula. It's taken this formula and it's gone to the ribosome, showing the transfer RNA. Here's the code. Transfer RNA, many of them are in the area, and inside the cytoplasm, many amino acids. Where do the amino acids come from? From the very proteins that you eat. In digestion, they break them down into amino acids. These amino acids are in the amino acid pool, and transfer RNA picks them up. Different transfer RNAs will pick up different amino acids. Just like if you want to go on a taxi from point A to point B, you can't just climb onto any taxi, Luni, can you? You need to know which taxi goes where, or which train goes where for that matter. So in the same way, a different amino acid will go onto a different transfer RNA. And transfer RNA will then combine with the messenger RNA, if they were lying like this, and if that was messenger RNA, transfer RNA will combine in threes as well. And how? Based on, if that was A, C, C, then obviously that should be U. Remember, in DNA that would have been T, but now U, G, G. In that way, according to the base pairing. And they bring the amino acids and put them next to each other. These amino acids combine, and eventually they become proteins. And this is known as translation. Why is it called translation? Because we had a code in a particular format. This code got decoded. It got decoded and formed something else. It translated one language to another language. It translated the code into a protein. In that way, we have a protein forming. And the way these sequences are will determine which amino acids. The amino acid sequence will determine the structure and the function of the protein. With that, Luni.
All right. Mindset is we're going to take a break, but I'd just like to read you guys something from Pakihero Mafatle, and it goes, I am Paki Mafatle, a proud mindsetter. I was doing my matric last year and got 4 distinctions and made the Free State top 100. I would like to express my gratitude to everyone behind Mindset. You made a difference because you are so caring and thoughtful. Thank you, Mindset. May you continue helping more people that are less fortunate like me. I love you all very much. Furthermore, I'll be leaving to study medicine in Turkey, so I'd like to say goodbye, everyone. God knows I only have love for Mindset. Special thanks to Aslam for his classes.
Oh, wow.
Thank you and well done, and we love you too, and we wish you everything of the best, Paki, and safe travels to Turkey.
Oh, well done, Paki.
We're glad that we
all
made
the best.
a difference.
Keep in touch.
Yes, in your life. And with that, Mindsetters, let's take a quick break. We'll see you straight after this. Welcome back, Mindsetters, from that break. Shout out to all of you who are watching. So many of you. So many. Aslam.
Thank you, Luni. Guys, obviously we've gone through the summary of what we need to know in this section. Now we need to see how these things will appear in an exam paper, so we can then ask ourselves, are we on track? In other words, this session, and also at school when they give you homework, unless the teacher is just telling you to rewrite the textbook, in which case you must politely tell the teacher that this is not the way to do homework. The homework should be in the form of questions that will test what you have done. Where there are certain cases where you have to summarize other things, that's a different story. But mainly, our focus, especially at grade 12 level, is to sharpen your skills to write the exams, because that is your end product at the end of the year. You need to answer the question. And in order to get a good symbol, you need to understand your work first. So first is the description, the lesson itself, and we're teaching it, and then you're asking the question. So you should be sharp when it comes to questions. And in your possession, you should at least have 3 to 4 past papers. Remember, in life sciences, the papers have been switched around. Paper 1 from last year will be paper 2 at the end of this year with environment. Paper 2. And paper 2 that was written last year will now be paper 1 in the caps. So that's all you have to, but the content itself hasn't changed, except for human impact. It's been replaced that whole environment section. You're only doing human impact in environment. You're not doing population studies and social organisms and community structure. You're not doing all of that. You're only doing human impact. But all the types of questions you get are going to be in the same line, okay? So you must remember that. So starting with our question now, tabulate the differences between DNA and RNA. So first of all, we draw a table. When it says tabulate, skill, drawing the table. So in this particular case, you'll notice when they ask you tabulate questions, they always give you an odd number these days of marks. The 1 mark is to draw the table. And the question is given to you, DNA and RNA. So all you do is you draw your table and you put DNA and RNA. So if this was a 5-mark question, you already have 1 mark just for drawing this table. Now that's a skill that you need to carry with you to the exam. You don't have to study for the skill. You have to have the skill. It's 1 mark. So, and when we're looking at this, we're talking, in an exam situation, we would say the structural differences. But here the question is saying tabulate the differences between DNA and RNA. We'll start with the structural ones because that's the important ones, and in the end we'll add others, okay? And notice when you're drawing a table as well, you will have that lines drawn there to show that it's a table. And when you're comparing, when you're talking about something here, what you're talking about on the next side must be relevant to what you're talking about here. Even though it may be correct, but it's in a wrong place, you won't get the mark for that particular one. Okay? So let's start with that. The first one we learned is that DNA is a double helix, and RNA is a single strand. That's the first difference. Second difference is that DNA has deoxyribose sugar, and RNA has a ribose sugar. Third difference from what we learned, DNA has ATGC, adenine, thymine, guanine, and cytosine, whereas RNA has A, it has G, and it has C, but it also has uracil in the place of thymine. And I just want to do something here just to show you the difference. So let's use pink there. So there's T. So T has been replaced by U in RNA. Mainly found, now we can go for the other differences, right? Mainly found in the nucleus and sometimes in the mitochondria and chloroplast. So now we're talking not structure, but we're talking about location. And here it's found in the nucleus and cytoplasm. We can even say in the ribosome. But remember that the ribosome is found in the cytoplasm. Just remember that. Okay? Yeah, that's about the important ones. I'm not going to go into other small niggly differences between DNA and RNA. These are the chief differences between DNA and RNA that you need to know. Sometimes you must also understand there is a document out there called the exam guideline that has been distributed last week to the schools. Please ask your teachers to provide you with that document. That document tells you this is the work that we are doing. This is what is examinable from the work that we're doing. So you need to focus on that before you look at extra. It's always nice to look at extra, but you must first focus on what is important, and then you can do the extra things, okay? Okay, so that's our answer there. Notice what I said earlier, double helix, single strand. Now, if I wrote here double helix and here I wrote ribose sugar, although ribose sugar is correct, but it is not linked to double helix. So you'll get this mark, and this one here will be marked wrong. So remember, you must keep the comparisons that are relevant to each other next to each other. And you must draw the table, obviously. Okay, good. That's the first one done. The other way that they will ask you DNA and RNA and protein synthesis is obviously the diagram below represents protein synthesis. Study the diagram and answer the questions that follow. So they're giving you a diagram showing you the whole of protein synthesis. And again, what I told you last week about a diagram is easier to learn from than from chunks of text. If you remember a diagram like this, or you draw your own diagram to show DNA, to show RNA, to show replication, to show protein synthesis, then that diagram is the one you're going to think about when you're writing. And simply from that diagram, you're going to then carry over your work into your notebook or in your test for that matter. Okay, so keep that in mind. So here they're giving you a diagram. They're showing you that this is the nucleus because the clue there is nuclear membrane, obviously. And the outside the nucleus would be the cytoplasm. This you should know from grade 10. They're showing you there's something there. What is this? The shape tells you what it is. That would be DNA. And they're telling you a process is taking place here, which has resulted in a single strand structure. Okay? They're also showing you these guys here, and they're telling you what those guys are called, tRNA or transfer RNA. And they're showing you this organelle here, and they're labeling it as C, and then they tell you there's the outcome of this whole process. It's a protein that has formed. So I've just gone through the diagram just to analyze. You need to obviously look at the diagram because it's a data that is provided with the question. But you'll do it much quicker, but you're going to scan through it. And make notes on the question if you have to. Let's see what they're asking now. Name the following processes, A and B. A is taking place where? In the nucleus. And that would be transcription. And B is this process here happening here with this transfer RNAs, and that has to be then translation. Describe how the messenger RNA is made from the DNA template during process A. That's already a giveaway to the first question, isn't it? Messenger RNA is associated with transcription, not just messenger RNA, but is made. Messenger RNA is also in translation, but it's not made in translation. It is providing the code during translation. So that's why it's so important, guys, to read the question, because sometimes the clues are there. You just need to focus. Remember, in exams, you are nervous, etc. First and foremost, you've got to relax, but you need to know your work. You can't relax if you know nothing. Huh? You need to go there prepared. proper planning prevents poor performance. Okay, how mRNA is made from DNA? So that we have to describe transcription. When does it happen? DNA replication, and I'm going to summarize it. DNA replication takes place, right? While that is happening, you don't have to describe the whole of DNA replication. You say while DNA replication is taking place, messenger RNA nucleotides. Remember from last week, a nucleotide is phosphate, sugar, nitrogenous base. It could be phosphate, sugar, adenine, phosphate, sugar, guanine, phosphate, sugar, cytosine, and in RNA's case, phosphate, sugar, uracil, or in DNA, phosphate, sugar, thymine. These are nucleotides. I mean, these guys are parking there in the nucleoplasm. And while the DNA unwound and separated, unzipped, now it has exposed nitrogenous bases. So what happens there is when this exposure takes place, A, C, G, T, A, when that exposure takes place, instead of DNA molecules coming here, RNA molecules come quickly into this area and they sort of steal the code. So here A is supposed to combine with T in DNA, but there's no T in RNA, so that will become a U. C with G, G with C. T, can we now make that a U? No, T wasn't supposed to be on the other side. T combines with A normally, and there is A adenine in RNA. So you don't make that a U. The U must come where the T was supposed to be, okay? And A would become U again there. So we'll have this messenger RNA moving out there. So that's what's happening there. The messenger RNA nucleotides combine with DNA to form, I would rather say RNA nucleotides, to form messenger RNA. This messenger RNA then separates from the DNA molecule and leaves the nucleus and goes to the cytoplasm and becomes attached to the ribosomes, exposing the codons. The codons are the base triplets. Good. Write down, I didn't write everything there, right? Write down the numbers 1 to 3, and next to each number, the nitrogenous bases that will complete the table. I'm going to do it on the table for you. We're not going to write down. In the exams, obviously you're not going to rewrite this whole table. You will write 1, 2, 3, and you will write your story there. Remember that. You don't have to redraw the table. The instruction told you what to do. So here, number 1, look at what they're telling you. That this is A. This is the first one. On the DNA, it's CAA. So what will it be on the messenger RNA? They want to know. So for C, you will have, let me put it in yellow, you will have G, and A, you'll have U, U. So G, U, U. And what you'll find on the other side, on the transfer RNA, remember that the messenger RNA is opposite to DNA and transfer RNA. I'll explain something just now as well. So if that's G, that becomes CAA. Notice in this case that the DNA and the transfer RNA are the same, because both of them are opposite to messenger RNA, anticodon. Okay? And here they gave you the messenger RNA. You need to work backwards. If that's G, that will be C. If that's C, that will be G. And if that's A, that should be U. And notice again, CGU, CGU. The only time that will be different, the only time that these two will be different from each other is if in this DNA you have an ATT, for example. So ATT will become UAA. And in this amino acid, that will become A, still the same, but now it doesn't have T. So it will have U and U. In that case, only for the difference of thymine, because that's the difference between DNA and RNA, there's no thymine in RNA. So that's the only time we difference. So think very carefully. The DNA and the transfer RNA are generally the same because they are both opposite to messenger RNA. Huh? It's like the patty in the middle of a burger. The two roles are the same. The patty is different. Don't write that in an exam, please. You won't get any marks for that. The questions below are based on protein synthesis. Describe the role of DNA during protein synthesis. We've done that already. Or rather, DNA. All right, describe the role. What is the role? DNA provides the code for the formation of messenger RNA. How? During replication, a strand acts as a template. What's a template? It provides the mold for another strand to form, and that's how it works. Notice the important words, the code, template, and during replication this is happening. The diagram below shows the sequence of nitrogenous bases of a small part of a strand of DNA which codes for the part of a protein molecule. So here's sequence of nitrogen of small part of DNA, not RNA, but DNA. So they're giving you the code there, CGG, TAT, CCT. If they didn't tell you DNA, and they tell you this is of a molecule, why must I tell you now that it's obvious that that's DNA? Why? Because it has T. RNA does not have T. Remember that, that's something that you need to know. Okay, but let's take our big scribbles out there. All right. So we have that situation there. Write down the messenger RNA codon sequence that reads from left to right. Now in this case here, you put your number down, watch it, 3.2. You're reading from left to right, and always keep them in threes the way they are given there. So this will be G, C, C. That would be A, U, A. Remember, the T doesn't become an A because the T is going to combine with A, and A, there is in RNA. Okay? And the last one, CCT, will become G, G, A. And with that, Luli.
Alright, Mindset says we are going to take a break. Quick shout out to Sean Perkins, Tatoyaune, Vianney, Takani, Kayam, Kanyisa Buhle, Tapedi, Dumelo, Marakeng, Mpo, Stoli, and yes, Mpo Stoli is the last one. Thank you so much, guys, for tuning in. Let's take a quick break and we'll see you straight after this. Welcome back, Mindsetters, from that break. I hope you guys are still enjoying the show and you're having fun with Aslam and I. So, Aslam, yes.
Are we there again?
Yes.
Back to Aslam. Okay, the next question talks about a table below shows the transfer RNA. Now notice again, they're talking of the transfer RNA anticodons and their corresponding amino acids. So here's a table showing you the transfer RNA and its corresponding amino acid. Okay? Again, this table will always be given. Select and write down from the table above the amino acids in correct sequence that would be required for the base sequence of the, I'm going to change the question, for the DNA template above. Which DNA template? We had one there on top, remember? Which one was that? This one here. So that was CGG, CGG, then TAT, and CCT. Okay, so this was our DNA. Okay, we want to know in that correct sequence which are the amino acids that were coded for by this DNA. So it was CCG. It's easy because the transfer RNA will be the same. I'm hoping I chose the right ones. I didn't because this one here doesn't have a CCG. Okay. Remember, I just changed the question because there was a messenger RNA there. Let's make the CCC. Then it would be that one, and that would be glycine. Remember, in that case. But this is an interesting one, and I'm hoping with my big mouth that I got this one right. I don't think so again. TAT, let's make that TTA. Let's make that TTA, all right? Let's take that out, TTA. TTA is equivalent to what on transfer RNA? It would be UUA, am I right? Yes, I am. So UUA is that one there, and asparagine actually, all right? And let's see if we have the other one, CCT. CCC is also not there, so let's make that CGT. CGT will be equivalent to CGU, and that would be alanine. What do you do in this thing? All you are doing is you are matching a base triplet with what the table, but you need to use your skills. If they tell you transfer RNA and they gave you transfer RNA, then you simply choose from here. But if they tell you transfer RNA and they give you DNA, like I've just done, then you need to convert that DNA to transfer RNA code. If it's C or G only, or C, G, and A only in it, then obviously it's a direct replacement. But whenever there's a T in the DNA, that T must be replaced with a U. Okay? Only in that case. If they gave you a messenger RNA, let's take the same example. We had CGC there. So we would have had GCG. Okay? So this is messenger RNA now. And the other one we had CGT, so that would be GCU. GCA. Sorry, GCA. Because A combines with T. And the last one I said UUA, so that would have been AAU. In a messenger RNA, you're going to get the opposite end coming. GCG becomes CG, where is it now? That one there, yeah. Okay? Not this particular one. Remember, I changed it. It was all C's. So that will be GGG, but there's no GGG here, right? Okay? Well, it will become CCC here. GGG. If that was the messenger RNA given to you, then you convert that. What is the anticodon? This is the codon. The anticodon would be CCC. And there's your CCC. Then GCA would be CGU. So you come here. Here's your anticodon for that. And AAU, is that a U? Yeah, it can't be a Y. There's no Y there, right? Okay. AAU would be UUA. And you come here, here's UUA. So that's what you're doing. All you are doing is they are testing your skills on base pairing. Remember that. Okay, let's move on. The manufacture of proteins involves an elaborate sequence of events. Describe this elaborate process in a mini essay. Now here they're talking about a mini essay, and they're talking about the process of protein synthesis. Okay? They don't say protein synthesis directly, they say the manufacture of protein, and that's what protein synthesis is all about. You need to write this in an essay. So first of all, what is the story on a skill? The skill we have to learn is find the verb. The verb is describe. What comes after this verb? This elaborate process. Which process? The manufacture of protein. So in other words, describe protein synthesis. And how do we do that in an essay? We simply start by saying transcription. We explain transcription, and we don't have to do it again. We've done it already today. They tell you that the double-stranded DNA unzips. That means replication is taking place. One strand is used as a template to form messenger RNA using RNA nucleotides from the nucleoplasm. The coded message for protein synthesis is thus copied onto messenger RNA, which moves out of the nucleus and goes to the ribosome. So you're explaining, you can add here that enzymes control this process. Okay? Translation. transfer RNA collects the amino acid and it becomes arranged on the messenger RNA according to the base pairing. And here you can even mention what base pairing means, A, U, C, G. So you're telling the examiner that I know how they pair. Then the anticodons on the transfer RNA match the complementary bases and amino acids become attached, and so you have it. Notice you are giving a point and you're finishing the whole of transcription, then you're doing the whole of translation, and that's answering the essay. Each line there is worth 1 or sometimes 2 marks. It depends if there's 2 important keywords used there. And this is how you explain the 2 processes, transcription, translation. They can add to this DNA replication in more detail if they want to in an exam. Okay. Going back, we go back to our challenge question. They say the first triplets of nitrogenous bases that form part of the gene, blah, blah, blah. How many of the following are coded for in the DNA template? How many nitrogenous bases? How many have we got? 1, 2, 3, 4, 5, 6, 7. How many nitrogenous bases in each? 3. 7 times 3 is equal to 21. That is simple arithmetic, not maths, simple arithmetic. The next one says, how many different types of transfer RNA? Now you have to be careful. Let's look at if there's any repetition here. That's not the same. No, 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 no. These 2 are the same, yes? And is there any other 2 that are the same? No. There's 2 are the same. So therefore, obviously the transfer RNA that carries this one is the same taxi. So it won't be 7, but it will be 6. And how many amino acids? They didn't say types of amino acids. How many amino acids? If there's 7 codes, there will be 7 amino acids. Remember, if they said type of amino acids, it will be the same as that and it becomes 6. Here also, if they said the types of nitrogenous base, that doesn't become 6. Don't be silly now. It would be 1 is A, 1 is C, 1 is G, 1 is T. So there will be 4. I'm just trying to expose you to the different types of questions that you can get in this case here. All right. Write down the messenger RNA sequence from triplet number 4 to triplet number 6. We go straight there. Triplet number 4 to number 6. That means these ones. So if that's TGA, that's the DNA, the messenger RNA for T will be A, C, A combines with T, but there's no T here, it'll be U, ACU. G, C, G, C, A, U, C, G, T, A, C, G. That's your answer there. And one more question there was, using the table below, determine the amino acid sequence coded by triplet number 4 to number 6. They gave you the anticodons. Now let's go back. Let's go back there. It's TGA and GGA, yeah, GGA and CTC. Okay, so that was a DNA. TGA can't have T, so that will become A, not A, sorry, UGA. Going here, UGA, here's our guy. So the first one is threonine. This one here, from the DNA, GGA remains GGA because there's no T there. GGA is this guy here, and that is proline. And remember, you must give it in the order that they're appearing there, right, when you're writing it down. And the last one is CTC. There's no T, so it's CUC. That one there, and that is glutamate. So that was number 1, that is number 2, and that's number 3, according to that table. How many of you got that right, I wonder? Okay, so that's the answer to that one. Notice it's just a matching game, but you need to know your base pairing. Looney, any serious questions?
Yes. So from Matome, where does the amino acids that are picked up by transfer RNA come from?
Where does the amino acids that are picked up by the transfer RNA, where do they come from? The amino acids are found in the cytoplasm. Remember, when digestion takes place, you break down all the proteins into the amino acids. These amino acids goes into the blood system. From the blood system, they diffuse into the cell. In the cell, they are in the cytoplasm. So just as the transfer RNA are in the cytoplasm, these amino acids are also found in the cytoplasm, and they're picked up by the transfer RNA.
Okay, and then from Kayam, can Aslam please explain a point mutation?
A point mutation, although it's not in the syllabus, let's say we have A, C, T, G, A, G, C, T, C. If for some reason this A is replaced with a C, nothing else changes. So one nitrogenous base is replaced with another. Because it's happening at one point, we call this a point mutation. And if you want to learn the other one, the frame shift, if we add something in between, if we add another T here, that means these will be the three letters, then it will be TGA or TGA, then GTC, etc. Frame shift, the whole frame shifts. By the way, it's not required at this stage in the syllabus. It's not in the exam guideline. Remember that.
Okay, and then from Dumelo, is it right to say that nucleotides are found in the nucleoplasm?
Just 1 second, Luni, before we go there. Just remember, guys, however, a mutation is any change in the gene. If the gene mutation is any change in the gene. Chromosomal, in meiosis, we will learn if a chromosome, there's a problem on it. Yes, you wanted to say.
All right, Dumelo, is it right to say that nucleotides are found in the nucleoplasm of the
nucleus?
Yes.
Tumelo, that's correct. If it's messenger RNA, the RNA nucleotides and DNA nucleotides are found there. And that is where the manufacture of the messenger RNA takes place.
Okay, and then this is from Progress. Hey guys, do I really have to know all the amino acids that are involved in protein synthesis?
Progress, you are not making any progress because you're not listening to me. I said that twice on the show today, right? But maybe your question came before I said that. Remember that all of you mindsetters, you do not have to know the table. If they ask you questions with the table, they will always give you the table. Your job is to know how to match DNA with messenger RNA or transfer RNA. From there, you match it on the table and you choose. Selection only.
And then from Tapedi, what is zona pellucida?
What is?
Question. Zona pellucida.
Oh, zona pellucida, that we'll talk about under sexual reproduction later.
All right. That's it now.
Is that all?
Yes.
Okay, guys, you know what to do now. You need to know the RNA, the types. You need to know where they're found. You need to know their structure. You need to know transcription, translation, and you need to match, mix and match according to base pairing at Cute Girl. Luni.
All right. We love you. Thank you. Thank you, Aslam. We will see you next week. Goodbye.